All right, everyone. Yeah, good afternoon. Thanks for joining me on this nice Monday. It's a little gray here in Germany, but yeah, I want to take everyone out to the tropics this afternoon, just for at least an hour. Get out of this windy northern Germany weather. So yeah, my name is Henry Wu. Thanks for the introduction, Raina. Germany, working on the Make Our Planet Great Again German research initiative project called Project OASIS. I'm currently at the Leibniz Center for Tropical Marine Research. And I first want to acknowledge the funding agency, Ministry of Education and Research, the BMBF, under the MOPGA GRI, and implemented by the German Academic Exchange Service, the DAAD. So as you know, I'm at the ZNT at the Leibniz Center for Tropical Marine Research, but I also have my partners at Lamont Doherty Herbary IRD for samples, and then for technical assistance with analysis at the Alfred Wegener Institute and at the Room. Okay, so my project is called OASIS. The full title is Witnesses to the Climate Emergency, acidification crisis and global warming from tropical corals. And you can get the O and the A and the ISS everywhere else. So I chose the acronym OASIS because corals in the tropical ocean is really a sort of the analogy to a desert, right? That it's nutrient poor, but yet it's teeming with life. And so it's on this basis that I started my project thinking about how we can best observe what has been going on in our tropical oceans. And so as you all know, that increasing atmospheric CO2 from man-made climate change is warming our oceans. And what I'm showing here is the original hockey stick curve on the left, the Mann et al. 1999 study that was published in GRL. You can see this is the hockey stick reconstruction featured in the summary for policymaking in the IPCC third assessment report, 2001. And then on the right is the newest one from the IPCC sixth assessment report from 2021. And you can see stick is really on for human caused climate change. And it's 2 decades later that the hockey stick you can see here, it ended in 1998. The stick has just gotten longer, or the hockey blade has got sharp longer, and it's no longer just a northern hemisphere record that was published in 1999, but really a global signal. However, as you can see, the shading of the temperature reconstruction here is the uncertainty, and it was even larger in the past. So in order going forward, we need to really better constrain some of these older records, and we just need more data. And so part of the requirement needing more data going back in time is that historical records from the ocean, especially in the tropical oceans, is really sparse. And without the historical SST measurements, we require to really knowing our past to model the future, and also to know what has happened under natural climate change. As you can see here on the left is the ICOAD's data record going back in time from the 2000s all the way to the early 100. It is the International Comprehensive Ocean Atmosphere Data Set, and it is the most extensive collection of surface marine data out there. And as you can see, for our Fiji Tonga grid, Fiji Tonga Rarotonga 3-grid area, grid as in degrees longitude, about degrees. In the most recent decades, you have a few hundred observations, but going back further in time, past the 60s, you really drop off down into nothing. And you have a few observations, and then the wars, kind of hit through and you really have you have a significant drop off. And this is a larger grid size across areas where you guys actually have pretty good records, but yet during the wars, they significantly drop off, and really the best records start only in the 70s. So that's one problem that we have going back in time regarding the reconstruction of temperature. And what about pH? As I was here, the oceans take up about more than 41% of anthropogenic CO2 emissions, and this has really significant implications for shallow marine ecosystems. And besides the warming caused by man-made climate change, the atmospheric CO2 decreases the pH and has been projected into the year 2100, which is here on the right, to significantly decrease the pH and also changes in the aragonite saturation. And you think the year 2100 is pretty far away, but yet we're in 2022 already, and that's only 80 years away, right? So, I mean, you know, my grandmother is 80 years old. That's just one generation away from where our kids really will see the significant change in pH and carbonate chemistry changes in the global ocean. And so what is oceanification? Ocean acidification, CO2 dissolved in the ocean, and it really triggers this sort of far-reaching consequences where carbonic acid is produced and the water becomes more acidic, so the pH declines. Since the beginning of the Industrial Revolution, the average pH has decreased by about 26%. So it decreased from 8.2 to 8.1, but every 0.1 is a logarithmic scale change. And so it's changed by about 26%. And as the ocean acidifies, the concentration of carbonate ions decreases. And calcifying organisms such as corals, mussels, clams, they really would have a hard time building their shells and their skeletons, because the less carbonate ions it has, it would cost more for the coral or the calcifying organisms to become to make their skeletal materials. And other marine organisms also require more energy in a acidified world, acidified water. And these additional energy is sort of for the organism is like under survival mode. And you can see here on the left, bottom left, that as the CO2 increases, the amount of bicarbonate also increases, the hydrogen ion increases, but pH decreases along with decrease in carbonate ions. And so, as I was saying before, you have calcifying organisms under threat. You have coralline, algae, forams, corals, deep water corals, mollusks like oysters, clams, and you have lobsters, and all these other organisms that depend on the coral reef ecosystem to survive. And so my point here is that we really need a better prediction for future climate scenarios to understand what would be the changing environment for these animals. And one consequence of ocean acidification is the study I did back a few years ago, where we cultured baby corals, coral larvae, under different warming and atmospheric CO2 scenarios. So a few years ago, when we did the study, we did it at 390 ppm of PCO2, which at the time was the baseline, but now we're way above 400 already. And we also had a condition at 750, and we had two temperature conditions where we had 27C and 29, which is sort of not the higher range of a coral survival, but it's reaching the sweet spot or the curve where the corals become unhappy because the water is too hot. And as you can see, at 390 ppm of PCO2, at both temperatures, the baby coral larvae are growing nice and happy, nice and round, very symmetric, with a lot of good skeletal materials. But once you turn up the PCO2 levels to future climate scenario at 750 ppm, you really see that it doesn't matter what temperature for the coral, they are very unhappy and they start growing these abnormal skeletal features, and they're no longer so robust and so corals have the ability to keep all this climate and sea surface information in their skeletal materials that you see that they're building up here. So in this project, we use massive hermatypic corals as climate archives. So coral reef builders are these massive corals, and they grow like trees where they lay down these annual skeletal layers growing outwards. And so you see here is a sketch where the youngest living tissue is outside, and you have these annual layers of a coral growing backwards in time. And these corals are just sitting in the ocean and they're constantly filtering the water. And so whatever is in this water, they get filtered and it gets locked into the skeletal material as they grow. And so others have so many stories to tell in terms of what they survived, what they've seen, what they lived through, and sort of the environmental conditions that they've witnessed. And some of the things that we measure in these coral skeletal material is for sea surface temperature, for salinity, for photosynthesis, CO2 uptake, and as a pH. We also can reconstruct seawater carbonate chemistry. We also have seen other studies use riverine input, runoff, and nutrients and upwelling. But for this project, we're really only focused on temperature, salinity, pH, carbonate chemistry, and a little bit of the riverine input, the runoff, as a proxy of precipitation. Really not go deep into details on proxies and proxy reconstruction because talking about strontium calcium proxies or delta 18O really takes hours and it's a lecture. And what I can tell you is more on a basic level that let's take strontium calcium for sea surface temperature reconstruction, for example. Strontium is a 2+ ion, and so is calcium. Calcium is also a 2+ ion. And the substitution of strontium for calcium is temperature related. So when you have a warmer ocean, you would have less strontium getting substituted for calcium, and so less strontium incorporated into the skeleton, and vice versa. When you have colder water, more strontium gets exchanged for calcium into the skeleton. Calcium carbonate, CaCO3, you get strontium and carbonate mixed. So it's these ratios that we can see going back in time, the changes in sea surface temperature. And same with oxygen isotope, delta 18O, the notation delta, just the ratio of the isotopes 16 oxygen and 18 oxygen is the ratio. And so for evaporation, precipitation, you realize that when a water evaporates, the lighter isotope gets taken up first, lighter, 16O, versus the heavier isotopes sort of stays below in the water longer. And when you have precipitation, when you have rainfall, the heavier isotope comes down first. So you have more oxygen 18 raining down. And so the changes in these ratios allows us to reconstruct evaporation, precipitation, and also salinity based on the changes. And pH for delta 11B for boron is following the same similar concept as well with boron and uranium calcium for the rest of water carbonate chemistry reconstruction. So we start off this project by going out in the field. Actually, for this project, we didn't actually get to go out because of COVID. We had many field canceled in the early pandemic. And so we had to pivot into having a lot of our coral cores sampled by our international colleagues. And we also use previously sampled corals to proxies, something that we never done before, sort of like a sustainable way to reuse some of these coral cores, and we don't have to go out and collect new ones. And so you can see here, some of the corals that we use are massive. Here's a picture of one with a scuba diver scale, a giant from American Samoa. The locals call it Big Mama. And in a paper recently published in 2015, 17, I believe, this coral was dated back to 1520. So this coral is about 500 years old. So we send out a scuba team. Normally we send out a scuba team and we take out a coral core by drilling into the coral. And as we said before, only the outer edge of the coral is the live coral, and the rest are just skeletal material. So our colleagues would go out, we would sample the coral, and we actually would plug the hole back up. This is a picture of Big Mama a few years after the coral collection. And you can see here that some of the holes have already grown over by the coral itself. And the coral is still growing very well. And so our technique is really trying not to have leaving open scars on the coral so that boring organisms or other fishes cannot get in and kill it from the inside. And it would be a shame for old coral to be ruined from the inside. So once we have the corals retrieved, we have these coral cores, we ship them back to the lab, and we first cut them in half. It's a core. We cut them in half. You can see here on the left, we have a coral archive and a sampling. So we have one that we save for later analysis, and also for posterity, whatever it is that we need in the future, we can actually go back to this half of the core to sample or not. But side of the core half that we definitely do sample, and we take a lot of skeletal materials from it. And here you can see that these corals are broken up into little pieces because our core catcher is only 40, 50 centimeters long. And so there are these connections that we have to make sure fit together. Otherwise, if there's no perfect fit, there's a possibility of loss in time because what we're considering, the skeletal materials, you could have gaps in age, and it would mess up the chronology, and our temperature reconstruction, any environmental reconstruction is only as good as the fit of our coral cores to each other. Once we have the coral core, we would take an X-ray of the cores, and the X-rays really reveals these nice annual density banding pairs. So the corals produce these alternating banding pairs of a dark band and a light band. And the dark band and the light band are based on density, because these are X-rays. And so when you have a higher density banding, usually occurs in the optimal growing season when the coral is growing faster. So there's lower density banding growing in the optimal growing season during spring and summer, where the corals growing faster, that there's their linear extension, their density is lower. And during the winter months, they're growing slower, so their skeletal density is higher. And you can see here on one coral, the differences in growth rate. We have a Porites here on the left that's growing anywhere between 12 to 15 millimeters per year. And we have another type of coral that we use in this project, Diplomastria, that's growing only at about 2 to 4 millimeters per year. And these alternating density bands really tells you how many years and how old the coral is. And you go from the younger core to the older core. And we use these X-rays to figure out where to best sample the coral climate reconstruction for the proxies. So we try to normally follow a coral polyp down the vertical growth axes, so nothing on the side walls where the coral is kind of growing out to the wall, but really where the corals are growing the fastest. And we try to follow one single polyp all the way back down to in time. So here's a picture of our PhD student, Sarah, who was sampling one of the corals from Rotuma. And you can see here that we use these little micro drills at about 0.4 millimeter, and we sample these parietes at every millimeter growth rate. So these corals were growing anywhere between 12 to 15 millimeters per year. And by the way, we're sampling out every millimeter, we're getting more or less a monthly signal from the coral skeletal material following the X-ray down that we can say this is the best growth of the coral, and this is where we get our information of the condition that they were growing in. After we sample the coral, we send our skeletal powder into mass spectrometers to analyze for delta 13C carbon ratio or delta 18O for oxygen. And we also take 500 micrograms to analyze for on the ICP-MS for trace elements for strontium, magnesium, and we also use the samples from the annual averages to analyze on the boron for the boron isotope. So boron isotope analysis is actually really time intensive is done in a trace clean lab. So the entire lab is made out of plastic so that there's no metal because we're analyzing for trace metals, and that we always have to get dressed up, and it's very difficult to move around in some of these outfits and in these containers. And so we extract the boron from our coral material, skeletal material. So we have to first remove the matrix or the calcium, and then we have pure boron, and we analyze the isotope within using a multi-collector ICP-MS. And yeah, so I'm not really going to go into some of the details of the analysis here because that would take up a whole nother day. So now I'm actually switching gears and talking more about the project itself. So the original overarching goals of the project, we wanted to reconstruct pH and carbonate chemistry using corals and boron isotopes, and really have this global assessment of ocean acidification before and after the Industrial Revolution, and also to understand the natural versus anthropogenic influence in our pH. My project has different work packages, modules, but since COVID, we really had to sort of pivot and change our modules. So instead of focusing only on the South Pacific Convergence Zone, we also have samples from the Central Pacific. And then instead of just samples from the Indonesian fluid flow in Module 2, we also use samples from the eastern part of the Indian Ocean. And then not only the Caribbean Sea in Module 3, we also have samples from the Gulf of Mexico for more of a tropical Atlantic reconstruction. And all three of these areas have their own distinct characteristics in terms of CO2 uptake, in terms of sea surface temperature variability. Either it's impacted by El Nino southern oscillation, or that there are large-scale ocean atmospheric interactions in terms of the Indonesian fluid flow, and then also in terms of the Caribbean current driving the Atlantic meridional overturning circulation in the Caribbean Sea. And finally, once we have all our data completed, we want to do a model simulation comparison to our proxy versus model comparison. And so, as I said before, here's a map of atmospheric anthropogenic CO2 uptake by our oceans. And so the characteristics for the Caribbean Sea is that there's really a lack of data. You can see the white spot, but yet it is one of the heavily populated areas, and it has one of the highest rates of uptake of atmospheric CO2. And we have in the South Pacific and the Central Pacific, we have an area that is lower in uptake, but yet it is a region that is heavily modified by the El Nino, impacted by the El Nino southern oscillation and the decadal, Pacific decadal and interdecadal oscillation or changes variability. And we have a region here in the Indonesian through flow, which is the only low latitude interocean connector. So this is the only place where you have warmer and fresher Pacific water moving into the saltier and colder Indian Ocean. This is the only place on Earth in a low latitude, right at the equator, where the oceans are mixing with each other. And to also pivot, we also have one sample from the Andaman area in the Indian Ocean, sort of as an end member of our Indonesian fluid flow. So we have samples from the Pacific moving on into samples in the Indian Ocean. So now I will get into the first results from the Gulf of Mexico, Cuba, completed by my PhD student, Marie. Here I'm showing you just how good the seasonal cycle are in our coral record. You can see here, these are all annual cycles that we're able to reproduce, and it matches really well with the surface temperature. That is a gridded sea surface temperature from satellite records. And you can see here so the coral skeletal delta 18O is a monthly resolved record. So we have data for every single month. The coral analyzed here from the Gulf of Mexico was sampled at every 0.4 millimeters. So we were getting about every 8 to 10 samples per year. And the correlation factor for delta 18O to sea surface temperature is 0.8, which is very robust for this area. And you can tell by the annual cycles that there are certain time periods that it picked up very well. So even 97 El Nino here, La Nina. And so when we reconstruct this temperature from this area further back in time to the early 1800s, you can see that the oxygen isotope and strontium calcium show a significant warming over the last 160 years of about 3.7 degrees Celsius, or about 0.23 Celsius per decade. From the 1980s, you can see that there's a slight plateauing, or even using strontium calcium, there's a slight decrease in the warming. But we have to take this with a little caveat that oxygen isotope is not only a temperature proxy, but yet it involves precipitation, as I was saying earlier, that we are able to reconstruct the delta 18O of seawater, which is a proxy for sea surface. And see here that it's becoming not only warmer, but also fresher conditions over time. And this trend has been seen by other studies in the region. And could this be attributed to a decline in strength of the loop current or the Caribbean current, leading to the Atlantic meridional overturning circulation? That's to be determined. But at least we see this significant warming for now. And in the reconstruction for sea surface salinity, or delta 18O seawater, we compared that to sea surface salinity. And it's a really robust fit. And we also see in the two records together that there is a change in decrease in salinity over time, which could be due to increase in precipitation due to warming, and or possibly change in the strength of the Caribbean current. You might ask, what about the carbon isotope? And here we're showing a record from Costa Rica, from a master's student, Sarah, from Costa Rica, this region. But before we get to Costa Rica, I just want to first show you that carbon isotope in corals has been shown to really track atmospheric CO2. And that here's atmospheric CO2 level, and here are Atlantic sclerosponges, Atlantic corals, Pacific corals. And you can see that this has been, these records have been normalized. And so you just have to really pay attention to the magnitude of change over time since 1800. But really the significant increase in delta 13C here over time since the 1940s. Really, the rapid change happens around mid-1900s. And our record from Costa Rica going back to 1976 really shows this significant decrease in delta 13C, which is also known as this oceanic Seuss effect, or the changes in uptake of 12C versus 13C over time. And moving on to the South Pacific Convergence Zone, this is one of the areas with a permanent feature, a permanent intertropical convergence zone feature in the Southern Hemisphere with a seasonal precipitation shift, which is related to this salinity front moving from the northwest to the southeast over time, pacing with El Nino Southern Oscillation. And over time in Fiji and Tonga, you see these little double peaks where the rainy season impact is over time. And you can see from our first record from the South Pacific, from Tonga and Rotuma, that there is a significant warming in the South Pacific, averaging from 1.23 degrees Celsius to about 2.1 degrees Celsius change since the 1800s. This is a slightly weaker relationship at Rotuma, likely due to oxygen isotope relating to sea surface salinity. Otherwise, the reconstruction is pretty robust, or the correlation is really robust relating to temperature. And so we also analyze carbon isotope here at Tonga and Rotuma, and you can really see the decrease about 1 per mil of carbon here on the left axis, which is the same as what we're showing earlier from Swart et al. 2010, that there's really this significant change since the 1940s that really is pacing the increase in atmospheric CO2 uptake of the ocean. And we also analyzed for the first time boron isotopes from Tonga. We're not so sure what the record here is. There's still some work to be done. We compared the Tonga record to another record from New Caledonia. The changes are more significant or larger at Tonga. And it could be that the records are specific. So we have one from Tonga, which is using Porites, and then we have another record from New Caledonia using Diploaustria. And so the boron isotope could be species specific in the reconstruction, so we still have some work to do. And we are able to show that there is the ability of the boron isotope to follow the pacing of El Nino Southern Oscillation, or El Nino and La Nina cycles that would actually have an impact on the differences in our boron content. But the main trend of our boron record from Tonga also does not change, that since the Industrial Revolution, there's actually a more rapid increase in, or rapid decrease in boron isotope, or pH, versus a more sort of but more stagnant since the 1700s. And now take you back to the atmospheric CO2 story. In 2018, I sort of did this compilation showing the rapid decrease in atmospheric CO2 by 22 records in the tropical Pacific. Doesn't matter which species, it's actually a really robust record that you have a change in about 1 per mil. So I just showed you the Costa Rica record that was also about 1 per mil since the 70s. And here, I just want to show you that it is not due to volcanic activity, as some climate deniers want to say that, what about the volcanoes? That there is no more increase in volcano causing this dramatic decrease. And then that solar forcing or impact solar variability, its radiative forcing is also not changed significantly over time. And so really this is all man-made, and then that we have this track record to the ice core records as well. So keeping that in mind, we have record from the Central Pacific from Fanning Atoll that also has this 1 per mil decrease in carbon isotope. And we also have a warming in Fanning in the Central Pacific. Here's Fanning, here's Hawaii. So the records really are in coherence with many published records in this region. However, when we did the boron isotope, some surprising results came back that there's really not there's no acidification from fanning using the boron isotope record. However, there could be even a slight increase in pH from this region. And so we're yet to verify some of these results. We want to actually also calcium ratio for total alkalinity reconstruction and also dissolving organic carbon to understand more of the carbonate chemistry picture and not only pH, as this could be a signal that the coral is regulating its own pH despite the warming and the increase in atmospheric CO2. And now we're shifting more results from Indonesia, from the Indonesian through flow in the Makassar Strait. You can see from our recent, we do have a warming and or freshening signal from our coral oxygen isotope from the 1960s. However, in our most, so this record from the Makassar Strait is here in the northern Makassar Strait. And here we have a record from the southern Makassar Strait where we don't have rapid ocean acidification. We actually have only a change of about 0.1 per mil, which is less than the 1 per mil that's been showing from the Atlantic to the Pacific. So something is very different here. And also the warming or freshening is actually not there. From this region in the southern Indonesian through flow, we actually have a more cooler and more saline conditions. And it's still work in progress. We're still trying to figure out why we have some of these some of these trends. Surprised me actually, but we have boron isotopes and trace element records that's in the works, and we hope to have this because we tease out the salinity signal and have another truer, more reliable sea surface temperature proxy using trace elements. And so coming to the conclusions or preliminary conclusions, because we're not quite finished with the project yet, we see in the Caribbean a warming of 3.7 degrees Celsius, which is outpacing the Paris Agreement 1.5 target. We have South and Central Pacific warming of about 2 degrees Celsius. And these are all records that also could be changed based on more data when we get it. But the record so far has been pretty robust. We have historical ocean CO2 uptake across the Caribbean, South Pacific, and Central Pacific, but we don't see that in the Indonesian through flow. Could it be this interconnection that is driving the change in carbonate chemistry, or a larger salinity effect impacting the CO2 uptake? And so we have ocean acidification reconstruction using pH from boron isotopes across multiple regions are in general agreement. However, Central Pacific indicate a slight increase in pH, which is, waiting to be discussed. And finally, some of the next steps that we're going to do here for my project, we have some verification to proxy accuracy and sensitivity from these regions using different calibrations, different corals, different calibration methods. We do want to have interbasin comparisons because we have all these corals spanning more or less the same time period, and we want to see if there are possible teleconnections across different ocean basins. And we want to learn more about the longer term sort of lower frequency variability and having a more definitive answer on this natural versus anthropogenic forcing using series spectral and wavelet analysis. And finally, we will have some proxy to model comparisons and to understand how future climate scenarios using our baseline calculations. And yeah, I'll leave this here. Thank you for your attention. And yeah, I'll take some questions. Thank you.
Great. Thanks a lot for that, Henry. Really fascinating. Rainer has step in and coordinate the discussion since his connection's a little bit dicey today. So, hi,
Okay.
I'm taking over. And I'm just going to ask you a quick question coming from the biology side before I turn it over to the other questions in the audience, because as you know, I'm fascinated by the work that you've been doing. And I roped you into helping us with this biology paper that we're trying to write for the Frontiers for Young Minds. I think of your work as biological, but you don't have any measures of the actual phenotype of the plant or the corals over your cores. Did you ever think about measuring the cell size or any kind of morphological thing that's captured in those cores to associate with the chemistry? I know your focus is on chemistry.
Yeah, so we do. We're after after they've already grown through. So we do measure their growth in terms of density and vertical linear extension. We don't do any of the morphological analysis because we are focused on one single specimen and they're mostly cloned. And so, yeah, I guess if we are interested in that, we would do it in a different way and focus on short cores and just on the very recent 5 or 10 years.
Yeah, I think that's a fair response. But I'm fascinated by the idea that there's so much diversity even within a species. You're trying to use a species of coral across this giant global gradient. But what we study is how within a species you get adaptation, you get differentiation. And I just wonder how much of the biological response to environment affects the chemistry downstream that you're measuring. I mean, you've done some really nice ground truthing to show that your measures are tracking what other measurements.
Yeah, so there are a lot of noise when you compare. So a coral from just a few kilometers away versus one that's 100 meters away, corals are, yes, living animals, and they both, one could be just having a bad day and not recording the signal, or not behaving the way it normally does on a normal day. And so the main goal, a lot of these studies, is to see the reproducibility across an entire region. And so reduce the noise and having their, the signal. But yeah, then the only way we could do it is having more reproducibility tests and just analyze samples.
Yeah, that's always the problem with biology. Okay, I'm going to go ahead and turn it over to Alessandro, who has his hand raised.
Thank you, Christina, and thank you, Henry, for a really stimulating presentation. There's a lot here. I'm a bit hesitant because this one, like possibly a climate denier question. You pointed out how volcanic activity is obviously, and I agree, not the underlying cause, but I'm thinking about local effects. You showed Tonga, and of course you recognize Tonga has been in the news a lot the last couple of days. And I wonder to what extent the seawater chemistry in these particular regions locally might be affected by isotopic exchanges with freshly erupted magmatic material underwater. I was thinking of some of with the Indonesian straits. Indonesia's famous for its volcanic activity, of course. And I'm wondering whether contamination, if we can call it that, from freshly erupted material or ash that's deposited in the sea might, at least for a short period of time, actually corrupt or contaminate the signals, the long-term signals that you're trying to measure. Is there any hint of that even?
There's actually, we do see cooling events after volcanic eruptions. So we have evidence in a tubo from Philippines' most recent eruption. And we also, going further back in time, we do have some from Indonesia as well. A lot of the cooling and changes in salinity and temperature. And in terms of volcanic CO2 kind of deposits. Nuria probably can be better answer that. But the corals actually do have a way to regulate their pH up by a lot, actually, to combat that. And could it be possible? Yes. In terms of carbonate chemistry, we never actually studied that. That's a very good question, and we should probably
So
look
for
into that as
strontium,
well.
calcium, and boron, do you think there's a possibility as well? Isotopic exchange with
Strontium, no, but for boron, it could be possible. Because boron, we're actually, so that's why we're doing all the boron chemistry in these clean rooms. You know, if you have air conditioning filters, that could easily contaminate our samples. And so there's actually a lot of different ways we can get boron into these corals. And so that's why we actually specifically sample in slower growing coral skeletal material, and instead of the faster growing, because we wanted to make sure that the boron is in a ragnite lattice and not just kind of sticking out on the outside. So yeah, but that's a good question. I have to look into that. Possible volcanic effect on some of these proxies.
Thank you.
Right, especially for the carbonate chemistry. Thanks.
Yeah, thank you, Henry.
Yeah, Nuria has a
Yes.
question.
Hi, Christina. Hi, Henry. Very nice presentation. Very interesting and very clear. Thank you. And you know, I have a suggestion. You know, like you have told us about your past reconstructions using corals, and this is amazing. And you know, I am ecologist, and I was thinking if one of your study sites, if you are thinking of planning also to deploy, let's say, instruments, pH instruments, temperature sensors, and in order that maybe, of course, that the scale will be completely different, but maybe, you know, to compare like more fine resolution scale.
Yeah.
Based with your course and also these actual or present data.
Yeah.
And another maybe related question is that I am just, you know, guessing, and I don't know, but do you think that, you know, when corals, they bleach and they lose the symbiotic algae, I don't know if this process, if it has also some, you know, like signal, or it could have a signal in the skeleton. And also if you could check this over the past.
Yeah, so answer your first question. We actually have we have a second project from a PhD student that we're actually using corals from Puerto Rico. And Puerto Rico, NOAA actually has a buoy right on top of the coral that we sampled. And so this buoy has been collecting PCO2, pH for the last 6 to 10 years or so, about 10 years, at a daily, daily resolution. And so we are actually using this coral that was collected literally right under the buoy to using laser ablation to actually really understand the fine scale and reconstruct on a weekly scale the changes and have a better calibration between carbonate chemistry and PCO2 levels, you know, above the water and then in the water. So that's one from your first question. And the second question is, we also, wait a minute. Can you repeat the second part again?
It was about the bleaching.
Right. So there's studies that shown boron has the ability to show bleaching, and other studies have not. So there's sort of species specific, some yes, some no, but most of the time bleaching signal most apparent in carbon isotope record, because when corals lose their symbionts, they don't photosynthesize and they feed more. And so you are what you eat. And so there you have a significant change in carbon isotope and nitrogen isotope because they're eating more to sustain their energy versus getting energy from photosynthesis. And another proxy for bleaching we are examining right now is actually microboring organisms. So eudendoliths, so you have fungi, blue-green algae that actually grows into these corals during blooms. And so during these algal blooms, that's more warming, by degree heating weak standards, that they actually make more borings into the skeletal material. And by that, we can actually conclude that there was a bleaching event. But not all bleaching events react the same way across different corals. So it's like me and you, some corals are more tolerant to hotter waters and some are not. And so, yeah, it's sort of what a lot of paleoclimate people are looking for as well, for this proxy to reconstruct bleaching, but we have not found the perfect one.